Yes, and I'm Nancy Hynes, and I'm also a group leader at the Friedrich Miescher Institute in Basel, and my lab works on breast cancer and does some work on normal development as well. So we have a lot of overlapping interests. Okay.
So we are in Vegas in Switzerland, and Mark Twain said of this place, a Sunday morning in heaven is noisy compared to the quietness of this place.
And where I always tell my students, so don't be afraid to go and sit at a table with somebody who you might call a big shot, because they're just normal people and they want to talk to you. They're interested in science. So don't go off and only spend all your time with the other students, but this is your chance to talk to famous scientists. I was probably always interested in science.
So what made you go into science and not art or sport? If I recall well, you are somehow related to the great Thomas Edison. Is this where your interest in science came from?
So my thesis was about we were looking. So I happened to look it up because I was prepared.
Oh, that's
so this is my first paper. I don't think you can read the title, but it's from the Journal of Bacteriology, which was actually an American Society for Microbiology.
We were using yeast as a model.
Yes, and so it was about turnover of poly-A-containing RNA in Saccharomyces, so in yeast. And the history behind that is that I had a very young boss who must have done his PhD and postdoc in the RNA field. And when he got recruited to Pittsburgh, and this was where Mary Edmonds was working, and Mary Edmonds actually discovered poly-A-containing RNA. So discovered this extension of RNA with the poly-A on it. And so since people didn't know what that was good for in those days, we decided to see whether it had anything to do with the stability of the RNA. And this is what I'm working on cancer research. We were working on steroid hormones. And he was interested in how steroid hormones controlled the differentiation of cells. So how do these hormones control cellular differentiation? And there weren't very many model systems that...
That's not the cow, that's the boat.
This is a boat that maybe you're going to see behind us, yeah. And so he was working on the chicken oviduct, and when we started working there, we were looking at isolating RNA. Again, it was RNA, isolating RNA, and looking at turnover, at the number of molecules in a cell, and how they were, a particular oviduct, egg white protein RNAs, and how they were controlled by steroid hormones. So in a way, the system was easier than the chicken oviduct, because you had everything that you needed was in this LTR. And so we decided very naively, we said, we'll start working on mouse memory tumor virus. And we asked Heidi Diggelman in Lausanne if she would be willing to hire us, and she was. And she was working on chicken retroviruses, and at those days I thought every retrovirus was the same.
We understand it today.
I never heard the word mentor in those days, so we would be... Well, I mean, we had these discussions, so he would guide our science, but he didn't really come around and ask, well, what about your future, your plans for the next 5 years? Because it was easier in those days to move around and to get recruited.
After 5 years at the Ludwig Institute for Cancer Research, you moved to the Friedrich Miescher Institute in Basel, where you are currently a senior group leader. What attracted you to the FMI and to Basel back then?
Yeah, so when we were working in Bern, we got to know first Alex Matter, who was he was eventually the head of oncology in Novartis, but at that point he was just hired in oncology to develop kinase inhibitors, and so we got to know him when we were in Ludwig Institute. And when we were looking for a job after the Ludwig Institute, which unfortunately closed because of monetary difficulties in the stock market in those days, when we were looking, and this was in 86 or 87, it was 87.
That must have been like a big drama.
It was a shock because they closed 3 institutes at the same time. And so then we contacted Alex Matter to see if there would be anything interesting in Basel, and he put us in touch with Max Berger, who was the relatively new director of the Friedrich Miescher Institute.
In most breast cancer,
Yeah.
Can you tell us more about this work?
Yeah, so this work stemmed out of our interest when we moved to the Ludwig Institute, and we started working on human cancer. So one of our early projects was to take either primary human DNA out of primary breast tumors or cell line DNA, and to transfect that into you could either do this in epithelial cells or in fibroblasts. And then we did transfections of genomic DNA, and we had incubators filled with plates, 100, 200 plates, and every 2 weeks we harvested these plates, harvested the cells, and injected them into 30 nude mice. And then sometimes we got tumors. So when we got tumors, then we isolated back out the DNA from the tumors, cloned it. In those days, you use ALU sequences, so human ALU sequences to get out your sequence, and then we sequenced it, and we found out we had a RAS mutation, that was actually the first RAS mutation that was discovered in a breast cancer. And we worked, he called the ERB2. ERB1 was EGF receptor, so this was ERB2. And so I thought, well, EGF receptor wasn't amplified, or we didn't find it, so maybe the ERB2 will be amplified. And so this was one of those exciting times when you develop a blood and you see, my God, it is amplified. Wow. And there were we had a lot in this particular panel. We probably had, out of the 30, we had 10, I think, that had this amplification. And so then I teamed up with Alan Goldhirsch, who was working at the Ludwig Institute in those days, a pretty famous clinician. I mean, he's very famous now, but he was famous in those days too. And we had a look then at these parameters that we could get a hold of with this panel of breast tumors, the clinical parameters. And we could see so that we had some correlation then with the worst.
You also generated an antibody against HER2, right?
Yeah, so the antibody, the first antibodies were generated with Mike Waterfield. And so we collaborated with Mike Waterfield, and somebody came down from Bill Gullett came down from the Waterfield lab, and together we had a look then at whether or not the protein was overexpressed. Because of course if you have an amplicon, it doesn't mean the protein is overexpressed. And so we were using these antibodies, we were the first to show that you also had overexpression of the protein. And that's a paper I'm very proud of, that cancer research paper.
So then you worked a lot on signaling downstream of HER2.
Right. The favorite right now is the RET receptor, because really, together with Heidi Lane, who was working in Novartis, this is the RET receptor. We discovered that it's overexpressed at the RNA and the protein level, not due to gene amplification, but in a reasonable percentage, 40-50% of primary breast tumors. So we're working on the RET receptor now, very mysterious what it's doing, and what its normal role in mammary gland, and then in targeting it in cancer, in breast cancer.
And developing models of RET receptor.
Right, we are actually.
So you worked in metastasis.
Yeah, so why? I mean,
yes,
for me it's obvious to work on metastasis, because this is what kills breast cancer patients. You can remove the primary tumor, and what kills the females is the metastasis that can come up 10, 15 years later, can appear and then kill a patient, because we don't have any cures for metastatic breast cancer. And so this is the challenge in breast cancer, of course in other cancers as well, but I think it's because breast cancer is so common, and many women of course do survive beyond 5 years, so beyond the maybe treatment with chemo or even endocrine agent therapy, but then succumb to their disease 7-8 years later, and so how can we come up with newer therapies for metastasis? So this is why I'm interested in metastasis.
Important was this to you?
Oh, to see the success. I mean, this was really important.
And to train them or mentor them.
Well, this was, of course, a lot of, not only fun. I mean, sometimes at the end it was fun when they finished and they had a successful paper. Sometimes it was difficult, but I'm happy that most of the people who left my lab, they actually stayed in science. So they were that I somehow could and make them enthusiastic. They wanted to stay in science, and that there were many of them are quite successful, I would say.
That's rewarding.
Yeah. Yes. Yeah. So I think that it would be important to have more cancer centers that are there are more of them in the United States. Of course, U.S. is bigger, of course, but where you do have the opportunity to have the clinicians. You have a hospital, you have the clinicians, and these clinicians can have time off to do work at the bench or to run a research lab, and they interact with basic scientists who are interested in clinically relevant questions. And this is, I think, when you see what the medics have to do here when they're working in the hospital, they don't have much time to go off and do research. So we had some at the Ludwig Institute. We did have some oncologists who worked together with us, but they would constantly be getting a buzz, and they had to go off to a patient, and somebody had to finish the experiment. So they were torn between their patients and the bench. They didn't have enough time to do both.
But the idea is to create centers in which the two of them can interact.
Yes, and that the clinicians have more free time to free up some time for their
research.
For their own research.
Mm-hmm.
Yeah.
So
and breast cancer, but how you get the message across to the general public that taking something for preventing breast cancer, I mean, a medication that also has side effects, I think these are challenges.
So Nancy, what would you like to tell the next generation of scientists who will watch this video?
Yeah. Well, if you're curious and you like to discover new things, I mean, it's a great job. It keeps your people always say, "What are your hobbies?" And I say, "Well, actually, my hobby is my science.
My job is my passion.
My job is my passion." So, I mean, if you don't feel that way about it, then I would say don't get into it, because you have there are a lot of setbacks along the way, and so you have to be totally enthusiastic about it. So that I would say you've got to have this you've got to be driven